Go away, man.
Are we gonna do that first, or are we gonna go straight to lecture?
We're going to review some of the one of the questions that people got wrong. It was one of the more popular ones to get wrong. So I can walk you guys through the process. If you guys have been looking through the question and looking at your answers and you're understanding really why your answers were wrong, and that's great. But hopefully, if I'm going to go through this one question, I did it with all my other classes, so I'm going to subject you guys to it too. Right, that's pretty much how this is going to go. Yeah, we're going to do this one. Hopefully it'll give you guys a little bit better idea of how to approach the questions, maybe because this one directly from the test. I know I went over one to prep you guys, but this is maybe a little bit better just because you can be familiar with it. It is one that you guys all had. In case you guys weren't paying attention, the questions came from a bank. Each chapter had 10 questions in the bank, and then each person had 7 to 9 questions from each bank depending on the chapter. So most people had similar questions, but there were a few here or there that were different. They're also, we mixed up the number, like the question numbers, there's also, scrambled. And so were the choices. So like if you happened to have gone to somebody and said, hey, I got this on number 1, I put A, and then that wouldn't really help them very much because it was different for everybody. So that's what we're talking about. So first I'm going to make this chart that I know what we're all familiar with, but I think it'll help us work our way through kind of what is what. So this guy was viruses and prions, right? Oh, it's so delayed. It's like, whatever. Okay, and then the cell ones, we had way over here, we had archaea. I'm gonna put archaea over here on its little lonesome self. Actually, let's back it up just a little bit. Sure. That's a N and not an H. I know it looks like one, but eukaryotes. All right, honestly, you guys see how this chart is pretty simple. It's pretty basic. We might look at it and say like, yeah, no, like this is really basic stuff. How would that have helped me on the test? I swear to God, half of your questions could have been answered with this. And if you don't think I'm right, bet y'all come at me. Thank you. So, come at me. So if we look at this and we see that it is simple, and of course, yeah, okay, of course. Yeah, because it's not going to save on the slides or anything. So if you guys need to take a picture of it, that's fine. Because you know this is going to come back on the final, right? But so that's why I kind of wanted to go over. I'm not going to go over necessarily this whole chart because I know the chart makes sense to most of us, especially at this point, right? But here was the question that I wanted to go over with you guys. It was the one about if you had a patient sample that you wanted to figure out basically from just one test, was it protozoa, was it bacteria, or was it virus from one test, right? And then our choices were to, and if I remember correctly, correct me if I'm wrong about these because I don't have it memorized on the top of my head, but we were going to test for the presence of DNA. We were going to test for the presence of a lipid bilayer. I don't know why this is delayed, so I apologize that it's doing that. We were going to test for cells plus organelles. I'm just going to put organs. I don't have to write it all out. And then there was one more, which is flagella, right? Some people are reading way, way too far into things. A lot of people are not eliminating choices based on the fact that they don't understand one thing that has nothing to do with the choice, perhaps. So let me explain what I mean. So how would we do one test for determining whether it was a protozoa, a bacteria, or a virus? First of all, if we find them here on our little setup, we know that protozoa is a eukaryote. We know that bacteria is an eukaryote, but they're both cells, right? And then we know viruses are not cellular. You should know that. This is basic stuff. If you went into your test and you didn't know this, I have some news for you. You are not studying. Whatever you're doing, whatever that is not studying. It's just wasting your time, okay? Because this is bare bones basics. So if you couldn't walk into the test knowing that basic information, I don't know what to tell you. You need to go take a bio course before you come here or something, because this was the basics. And what we're about to get into, traditionally, like I've told my other classes, I'll tell you guys, I usually save the uplifting part for the end, but usually what I tell my other classes is simply that people do better on unit 2, and they do even better on unit 3. They do about the same on the final as they do on unit 3, typically. Okay, it's a little bit better. And I mean a little bit, I don't mean like they go from C to B to A, okay? I mean they improve. My averages do. And you guys did the best out of everybody. Your average was higher than everybody's by far, okay? To put it into perspective. So the average in here, I think I told you this was 75. Nobody touched it. Everybody, I think the next highest I got was 71 or something like that. So good job, honestly, with that. But still, I want to be sure that you guys do better the next go round, right? So unit 2 is going to be using a lot of this information, getting more complicated with it. Unit 3 is going to be all new, brand new information that you guys have never heard probably ever before. You may have heard what a neutrophil does, what a neutrophil is, and that it is important in maybe a CBC, if you even know that. It's the most numerous of all the white blood cells in a CBC if you're healthy. And in an infection as well, bacterial infection typically. But even if you didn't know that, you probably know nothing else about any of your immune system. You probably don't know what a lymph node is or what it does. We're going to talk about that in unit 3. But you're not going to be able to keep up with units 2 and 3 if you didn't get unit 1, which is just our framework chapter. This stuff, absolutely, I keep telling you guys it's going to come up over and over again. Believe me, it's going to come up over and over again. So if you think you can just set this aside and come back to it for the final, you're wrong. So get ready. because we're going to be moving forward today with genetics, which is one of our hardest chapters. So that's why I wanted to do this really quick. Since we're not doing Kahoot, do this instead. All right, so our question, how do we find out which one is which? So if we're looking for DNA, all we have to do is find one test that will tell everything individually, right? One test. So if it tells you that it could be these two, but not this one, that's not what we're talking about. It has to be one test that says it can be, it would be A or B or C, not multiple. So how many of these guys could have DNA? All of them, yes? Can we agree on that? So bacteria, any eukaryotes, protozoa, and then the viruses, all three can have potentially DNA, right? Some viruses don't, but that doesn't help you. All right, next is the lipid bilayer. Anybody know what that is? What does that mean in terms of the cell?
That's the membrane, that's where the permeable membrane is. So
Yep.
stuff can come in and
It's
out.
that lipid bilayer is your membrane. So do bacteria have a membrane? This isn't a stupid question. Like literally, do they have any membrane?
Yeah, it is.
Yeah. They have a cytoplasmic membrane, right? And a wall, and then potentially a second membrane outside of that. So they have a membrane. What about eukaryotes? What about protozoa?
Yeah.
Yep, they've got a membrane. Can viruses have a membrane?
No, they have that weird protein thing.
They can have a
Oh, yeah,
membrane.
they can. You're
Because
right.
if they bud through, right, they can wrap the membrane around themselves. And that's what they can. So that's called the envelope for the viruses, right? So anyways, so that doesn't help us, is what I'm saying. So not DNA, not lipid bilayer. So now if we move on to, actually I want to go to flagella because hey, in case you haven't figured it out, cells and organelles is the answer. So, but I'll tell you why in a minute. Flagella, can bacteria have flagella? All right, can protozoa, can eukaryotes in general, can they have a flagella?
Yeah.
And now one of the things we talked about was protozoa, they have 4 means of locomotion or non-locomotion, right? They have the flagella, they have the cilia, they have the pseudopods, or they can be non-locomotive, like not. So that's the 4 groups you can have with the protozoa that we went over in class. So those 2 already can have flagella. Even if you weren't sure about viruses, and viruses can't have flagella, by the way, they can't. I'll tell you why in a minute. But even if you weren't sure about that, you already know if you test for flagella, it's not going to narrow it down to 1 or 1 or 1, right? So you can just cut it out right there. You don't even know about the viruses. There's a similar question that I got a lot of questions about was about chloroplasts. What photosynthetic organisms would you find chloroplasts or not find chloroplasts in or something like that? And it was like plants and algae, and then there was fungi, and it was cyanobacteria, and then one of the choices was both fungi and cyanobacteria. And people were asking me about what are cyanobacteria? How am I supposed to be able to answer this question? Y'all, are you serious?
We talked about it in class, didn't we?
No, I mean, whether we did or not, cyanobacteria, right? Do bacteria have membrane-bound organelles at all? Chloroplasts, are they membrane-bound organelles? You see what I'm talking about here. It's not as hard as we're trying to make it out to be. It really isn't. It's not meant to be tricky. It's just meant to get you guys thinking about it in a different way. And I'm applying extremely simple information. And you guys are reading these questions and being like, oh my God, this is so complicated. How am I supposed to figure this out? But then I walk you through it here and you're like, well, shit, yes, thanks a lot, Professor Yost. Like, where were you when I was taking the question? All right, so we know flagella isn't, so we know cells and organelles is the answer. And one way you can think about this, look in a microscope at the patient sample. Does it have cells? If it has cells, it's not a virus. If you look at your patient sample and it doesn't have cells, it's a virus. Has to be. The other ones are cells, right? So we've made one step, but we're also looking at the organelles. So let's say you got as far as saying it's not a virus. We've knocked that one out. So now we're looking at our cell and we see membrane-bound organelles, even just a nucleus, which is a membrane-bound organelle. Any structure in the cell with a membrane around it that has a function is an organelle, right? So we look into our cell, it has any kind of membrane-bound organelle at all, it has to be the protozoa, right? It can't be the bacteria. Bacteria do not have membrane-bound organelles. That's one of their defining features. They don't have a nucleus. Guess what comes along with that? No membrane-bound organelles, right? So I hope, and like I said, I'm not trying to make you feel stupid. That's not my goal at all. I genuinely think you guys understand the material. It's just being able to apply it when you get into the test. Yeah. And I've already given you 2 examples of questions that you could at least eliminate some using just this basic kind of information. Also, one of the questions was asking about bacteria having mitochondria, something like that. No, of course they don't have mitochondria.
They call it the mitochondria.
basically, yeah. Okay, so that's that. That's me shaming you for this. Again, not me trying to, but I want you guys to learn from it going forward, okay? Use that as a learning experience. This is why I say everybody does pretty bad on the first test. Now you guys know what to expect. And figure out what you need to do as an individual to pick yourself up and move forward from this. Do we need to revamp how you're studying? Do we need to, find different study methods? Or maybe, you just have to admit to yourself, maybe crack down a little harder next time because you knew you were doing the right thing, you just didn't do it enough. Whatever you need. And I'm here to help you guys through that, but you have to come ask me. I'm not gonna, know that you need help with studying. I'm gonna assume that you've got it and you're figuring it out. One of my biggest pet peeves is people in my class who take this class who do poorly in this class. That, will hear me say this at the beginning of the class, and they'll do poorly on their first test. And they'll just be one of those people that are just adamant that what they were doing was fine in their other classes and it should be fine in here too. But that's cool, but you have to pass my class, not in Comp 1 again. That's not what we're doing, right? We're passing microbiology, not Comp 1 again, and so think about it that way. This is definitely a higher tier course than some of the other ones people have taken previously. There's a reason there's a 2 in front of it and not a 1. You guys know what that means, yes? It means sophomore level course. It does not mean baby now goes into the next step. We went from freshmen from the 1,000 courses into the 2,000 courses. These are sophomore level. You are expected to have a base understanding of stuff before you come to my course. So that's why I say, if you need help studying, you might, but that means the other classes before you failed you, likely, actually highly likely that the courses or schools or wherever you were before you came here probably failed you. I don't know if you've noticed, but since COVID, instructors have gotten very lazy. And they will just post crap online and just leave it to you guys and not even try to figure out how to help you learn it or anything. That's why I want to tell you, if you want my help, I'm here. If you don't, then that's fine. But if you choose to not have my help, not ask for my help, not ask for anybody's help, and keep doing what you were doing and keep doing bad on your test, it's on you if you fail the class, not me. Because people will complain about that in my evals at the end of the semester. That I was like, she never helped me do better on the test, and they never asked. I'm not gonna just do the work for you. So anyways, if you ask, you email me, or if you even want to be hey, is there some people you can get me in touch with for a study group or anything like that, then I'm happy to pass on the word for stuff like that. But whatever you guys need, you have to ask. Or even if you don't even know what you need and need help figuring that out, then ask me and we'll work on it to figure it out. But I say first go over your questions and figure out where you think you may have gone wrong. Like I said, categorizing it as 1, 2, or 3. If it's mostly ones, I think ones we said was just you knew the material, you looked at the question. Now, when you look at the answer, it makes sense without even really having to look anything up. That's probably just you need to study a little bit harder before the test type of thing. If it's 2, where you have to look up the information to figure it out, but you get it without having to relearn the whole subject, then that could be a different kind of study issue. You need maybe a little different a way to approach it. Maybe not just flashcards next time, right? Maybe now we'll also add in blah, blah, blah thing. But number 3 is a whole other thing. If you find that you didn't understand any of the questions, a majority of the questions that you were missing, and you had to basically go back and relearn and reintroduce these topics to yourself, then that is a matter of a learning issue and an overall big time study issue, right? So maybe we look at how you're taking your notes, what you're doing whenever you leave the class to go review the information that we went over on top of, what we need to do to actually study for the test itself. So that's a bigger question, and we definitely want to solve something like that before we get too far into it. So that, number 1, you can pass this class, sure, but I don't want you guys to have to go into nursing with no ability to learn the information effectively. And that's what I need for you guys, is to know that. Because another thing I told my students is remind you guys, it does not get easier from here. This is not the hardest course you're going to take. It's the hardest course. People say, oh, this is the hardest course you're going to take in your prereqs. Yeah. Nursing is hard. And you can go ask anybody that is doing well in it how hard they're working compared to their prereqs, and then come and tell me. Because I don't think anybody are going to say it was easier than micro. So I'm just saying. I want you guys to do well, and I'm hoping I can set you up for success. Okay, so if you have any questions about that, like I said, you guys did better. I want to remind you, you did better than the other sections did. Congratulations, you should be proud of that. There's something about Monday morning courses, man. They always do better. All right. Okay, so the nature of genetic material. We're going to talk about some terms that we need to know for genetics, some of the stuff is gonna be stuff we kind of already went over. That's why I went over it. So it would be a primer. So when we got into this, it wouldn't be too new. So genomes, anything in any genetic material in a cell or organism or however you want to look at it, virus, whatever it is, all of it together makes up the genome. So this would include not just like your chromosomes and stuff like that. Like we talked about bacteria, how they have their chromosomes where they can have little extra pieces like plasmids that carry extra genes. That's part of their genome technically as a whole. Any genetic material in there. Some can also be in non-chromosomal forms, like we were just saying, the plasmids we just mentioned. And then our mitochondria, or plants have their chloroplasts, or algae have their chloroplasts. They have their own DNA, like we talked about with that endosymbiosis theory. So yeah, but all of it together is the genome. And viruses can have DNA or RNA considered as part of their genome. Remember that our RNA is always going to be coming from our DNA, but theirs isn't like that, right? They potentially have RNA as their whole genome. So, all right. So a chromosome is any, it says distinct cellular structure of a neatly packaged DNA. When we see R's and those X's, and I think I said something about it in this class, but those X's, remember the X's are not what your chromosomes look like in your cell, right? That's whenever they're going through mitosis. That's when we have consolidated everything in our chromatids, really like shrunk them up so that everything's not willy-nilly all over the place, and we've made copies. So it's just a copy with a copy attached to each other in the middle. They call it the centromere. That's what the middle is. And then when we go through mitosis, remember how we're going to pull those things apart? Now the new cells have 1 and 1. So that's why they look like that. And the picture's easier to look at them in like a microscope and stuff when they're like that in the middle of mitosis. So that's why we're used to seeing it that way. But we have one linear chromosome per mom and per dad for each of our sets, right? So we have one from mom, one from dad for chromosome set 1, and so on and so forth for 23 sets. So we have 46 chromosomes, including your X and your Y. All right, so always in eukaryotes, when we have it, because we have a nucleus there and it's all membrane bound, all of your DNA, your chromosomes, proper chromosomes, are going to be inside of the nucleus. They can occur in pairs, which we call diploid, which is what we are. We're diploid, mom and dad pairs, or singles, which are haploid. If you were to look at our gametes, our eggs or our sperm, they are haploid compared to what we have normally. But some organisms can be just haploid in general, and they don't operate the same way that we do. We don't need to worry about it though. Bacterial chromosomes are usually, they only have one circular chromosome, and it is double-stranded DNA. It's just in one big old circle. All right, so your genes basically are going to code for one of 3 things. Structural genes that code for proteins, that's number 1. Number 2, genes that code for the RNA machinery that are going to be used for the first one. And then the third one, regulatory genes that control whether the first one is being made. When I tell you guys that literally all of your genes are for proteins, it's either the proteins themselves or the machinery that's going to make them. We're controlling them, obviously, but like, that's what your genes are for. All right, we can then look at somebody's genotype versus their phenotype. If you guys have ever heard of Mendelian genetics, or heard like the story of like the different colors of peas and how he would like cross the peas and like see what colors he could get or whatever. He was the father of genetics, basically. Gregor Mendel was. And the idea is, we know today that you can have dominant and recessive traits. That's probably not a new concept to a lot of people. Yes. Yeah, yes, that's exactly what I'm talking about. Yes. So we know we have dominant and recessive traits. I'm not really familiar with like some of the obvious traits, like which one is dominant and which ones are recessive, but let's just go with brown hair and blonde hair, like natural brown hair and natural blonde hair. I'm pretty sure brown is dominant over natural blonde hair. So let's say your mom is a natural blonde, okay? And she got 2 genes. Her dad and her mom both gave her their blonde genes, okay? So this would be mother. Well, let's say your father, he had genes, one from his mom and one from his dad. Both of them happen to have brown hair. Okay, and the big B means it's the dominant gene type. Okay, it means it's the one that'll get expressed over the little b. That's why we use it that way. So all of these, you have to have one from your mom and one from your dad, right? So you would bring this down or across. So we get big B and a little b, down and across. That's big B and little b. They're all, spoiler alert, they're all big B, little b. Okay, so that means if you have somebody that has two strong brown hair traits and they are having a baby with someone who even has two, recessive blonde hair traits, it wouldn't matter. All of their kids will have brown hair, no matter what, 100% of them. So that's how you can look at this stuff, okay? You can, so that person, say this is you were this baby that got the BB, you're gonna have brown hair, right? Because that big B is gonna be the dominant trait. It's what's expressed. Yes. It can be quite large. Yeah, I did that. Yeah, in my genetics class, we would have like these massive squares that we would work with several traits all at once. And it was really, it was kind of fun at the time, but don't make me do it now. I don't know. I couldn't do it now. But yes, so we have two terms that we can apply to this, your genotype and your phenotype. This, you have one gene from dad and one gene from mom is your genotype. So you actually are carrying this blonde gene, right? This person is anyway. This person's carrying a blonde gene, but they're just not expressing it. So that's their genotype. They have one brown gene and one blonde gene. So you could see if they were to have a child with somebody who also is carrying another blonde gene, you're getting a chance for having a blonde-haired child. It might just, we say skips a generation, but it's all genetics relating to that, okay? Who's carrying what genes. You can see how complicated this can get when we're talking about so many genes and so many variations of them. All right, so anyways, but they're going to be expressing brown hair. This brown hair aspect, what we see expressed, this is called the phenotype. So they're just two words here, genotype and phenotype. Genotype is the genes that you're going to have. The phenotype is what you actually see expressed. So even if you've been big B, your genotype would have been big B, and your phenotype still would have been brown hair, right? But so yeah, so sometimes your genotype can only be the phenotype, but sometimes you might have a hidden gene like we do here. So that's the idea. So phenotype is what's being expressed. Sometimes some areas of your body or some cells will favor expression of some genes versus others, just depending on what that organ is. And sometimes you can have a variation of those genes, turning them on and turning them off or whatever in different areas. If you guys have ever seen calico cats have this, and that expression of the different colors of fur on their fur, the different colors on their fur will be basically controlled by their X chromosomes. But you have to have two X's because it's a recessive trait. So you have to have two of them to see it. Okay. So anytime you see a calico cat, it's always a female. Every time.
Aren't there male calicos that are really rare that exist?
If they have genetic defects, maybe. I don't know anything about it though.
Oh, they might be intersex. Come to think of
They
it.
could be. I don't know enough about it.
Is.
But anyways, yes. So you get it. All right. So we already know our structure of our DNA. DNA, we've learned, is made up of nucleotides. Our nucleotide is our phosphate, our deoxyribose sugar, and our nitrogenous base. That nitrogenous base that we keep talking about, remember, that's going to be your A, T, C, or G. If on the test, I were to have you, recognize these, I'm not going to put, similar terms next to each other, not adenine and adenine or something like that, okay? I'm not going to do that. I don't need you guys to know that specifically, but I definitely would be aware of A, T, C, and G and what they're going to be pairing. G always binds with C, A always binds with T. And I remember this as G looks like C. So those are the ones that are going to go together. And then A and T both have some sort of horizontal line involved. They're the ones that are going to go together, okay? So, for example, the typical bacterial chromosome will have several million nucleotides. That's quite a lot. All right, so we say, this is a term that is absolutely going to be on the test, by the way, antiparallel. We say that DNA has an anti-parallel arrangement. This is because the backbone of DNA is running in opposite directions. So we know enough that we have our DNA helix and we can have a ladder basically. And we know how this goes, right? We've drawn this before, so we know how it goes. They're gonna be binding across to one another and whatever, so on and so forth. But this backbone isn't quite simple as that. So even if this is our nitrogenous base, the backbone, even though every one of these has to be attached to a sugar, let's say, then that sugar is attached in between to, do you guys remember what's going to be in between the sugars? This is the phosphates. right? Hold on, bear with me. Okay, the bottom might look the same to you if I were to draw out from out here, but the places where the phosphates are binding from one sugar and onto the next are at different places on the sugar. So what I mean is the different carbons. You guys remember anything about organic chemistry and how we number the carbons based on, whatever they're bound to? Yeah. So that's what this is going to be referring to, okay? One strand is going from the 5' end. So that would be like 5' would be the 5' carbon on your sugar here. And then this one might be the 3' carbon right there that's left out open on the end. Okay. So 5' to 3', and then on the other strand, you have the same thing, but it would be oriented in the opposite direction. Chemically speaking, they are literally like opposites of one another, going in opposite directions. This is incredibly important for everything DNA is going to do for us. So don't forget this. 5' to 3'. And then the other one is running, it's 5' to 3' this way and 5' to 3' the other way. Like they're going on sides of the street, okay? Don't think of it as one is going 5' to 3' and the other one's going 3' to 5'. No, don't think of it that way or you're gonna make it really hard on yourself. So they're 5' to 3' and like opposite sides of the highway. Leave it at that. Okay, so that's why they're called anti-parallel, because they're running parallel literally to one another. We're binding off the sugars, yes, the same way, but their backbone's oriented differently. So this is a picture of that backbone, how it's oriented. It's showing us our 5' and our 3' carbons. So this is our 5' carbon with this phosphate up here. And our 3' is down here on the bottom down here. And again, if we were to look down here, the 5' and then the 3' is here. So it's just talking about which one is at the far end of each of them. They're oriented opposite directions, like two sides of the road going each way. Okay, and then this is what's happening between our base pairs. When we have G to C or A to T, they get these hydrogen bonds in between them. Remember that part, how we're only being held together by hydrogen bonds? Well, this is where that's going to become important. Okay, so now we're going to look at replication of DNA. Replication of DNA is called semi-conservative. This means that we're basically going to go from we have our original 2-strand ladder, right? We're going to break it open. It's going to come up a lot later on too. We're going to break it open essentially, and then match things up to what's going on in there. So now you have 2, right? So we made a copy. That's easy enough. But these came from an old strand and an old strand, and we made new strands from those. So we don't have one that is still 2 olds and then a new one that's 2 news. No, it's going to be semi-conservative, where we have conserved one of those strands, and the new strand is made from based on that. Semi-conservative, right? So let's talk about this, because this gets really easy, right? Right, it already says not completely new. The new daughter strands are not completely new. One is old. And I'm not going to ask about which DNA polymerase does what. I just need you to know that DNA polymerase, we know what a polymer is. It's where we link our monomers together to make the thing that we're talking about. So DNA polymerase, ACE is always going to be an enzyme. So this is an enzyme that's going to polymerize DNA. We're going to make DNA, add on nucleotides, add on nucleotides, add on nucleotides, okay? So there's some things that we have to know about it. It has to have a template. It can only add nucleotides to an existing chain. It can't just like, if you ripped apart the whole strand, right? And it's just chilling here and it's not got any chain at all on the bottom of it, it can't start adding nucleotide. It has to have something here to build off of and attach to, okay? So it has to have some sort of what we would call it a primer. And it only adds nucleotides in the 5' to 3' direction. So this is why I'm saying to keep that straight, okay? Because 5' to 3', you're going to hear me say it enough. Hopefully it will just be drilled into your brain. It's 5' to 3'. It's 5' to 3'. But I'm not ever going to say it the opposite way. All right. So wherever, we can't just unbind all of our chromosomes all at once, right? That's nutso. Just too much going on there. So typically what we do is we start at a replication fork. We'll start at several places on chromosomes and like separate it out and then just start working from that point. We're just going out and eventually they'll all meet up with one another and we'll have made strands, but we have many sites where this is going to be going on. So that site, that single site where we're going to be having this going on is called the replication fork. It's where we're going to unwind the double helix and start separating those two strands. I drew them as completely separate, but it turns out they're still connected to the rest of the chromosome. That's all. Okay. They're still separating out. And each circular, if you were a bacteria and you were circular, you have two replication forks, kind of one at one end, one at the other, and they're going to meet that way. And then that primer, like I mentioned, you have to have DNA polymerase building off of something. We usually use RNA as our primer. Okay. And we'll go back and replace it with DNA afterwards. All right, so while we are going through DNA replication, and I think it went out today, the announcement that has the videos for genetics with it. I highly recommend that you guys go watch those because they're better at like showing you actively what's happening than I can draw, right? Because I can only do it like just stationary, basically. The videos kind of show it while it's happening. And there's a few of them I think that help with this chapter. But basically, when you're doing this, if you're synthesizing only in the 5' to 3' direction, this creates a problem, and I'll show you why in a minute. Because as you are opening up your little bubble, let's see if I can draw it on here. You got your bubble open, right? All right, and you started here, and you're going from 5' to 3', going this way. You can just keep adding nucleotides. Sure, this is your primer, whatever. And then you're going to keep opening up that bubble as you go along, right? Right, okay. So as that is going on, though, the other strand, you have to add it in 5' to 3' also, but remember, it's going in the opposite direction. So you can't start way down here and start adding on because that's all closed off. We have to open it up, right? So what's the problem here? Okay, well, good. That's fine. So what the problem is that as you open up this replication fork more and more, you're exposing more and more of this bottom strand that you can start adding on nucleotides in the, remember, it's the opposite, like we're two roads going in a different way, 5' to 3' direction. And then as you open up more, you add another little piece. Then you open up more, you'll add another little piece. And you'll just keep doing that as that gets opened up more and more and more, making little pieces. Okay, so the leading strand is the one in this picture where we can just keep going. We can just keep trucking. We don't need to make it in parts, okay? The lagging strand is the one where we have to open it up gradually and then make a little part, okay? Those fragments are called Okazaki fragments. And so they're made in that 5' to 3' direction on the strand that we would otherwise look at it and say it was going to 5', okay? So we're going, everybody has to be always going 5' to 3'. It cannot go in the opposite direction. It has to go 5 to 3. Okay. And since they're opposite, as we open it up more and more, that bottom one will have to be made in segments. Whereas the top one you can see will just be pushing along as you open up. Okay. The video is better at explaining it than I can draw it, I promise. But okay. It's showing all the little parts and moving parts and everything is what they're doing. But here's our 5' strand down here. Pushing right along, adding our little red nucleotides. I don't know why that line does that out to the side. Pushing along with our little red nucleotides straight along. This is our leading strand, right? And as we are opening up, we get more and more space to add in these little fragments, right? But like we have said, we don't build off of nothing. So each of these fragments has to have a primer, every single one of them. So RNA primer here. So this gets real complicated when we're talking about lagging now. The lagging strand, making those Okazaki fragments with the RNA primer. Then we remove the RNA primer, replace it with DNA nucleotides, because now we can attach to the other fragment next to it. Oh my gosh. And then we can seal them all together with an enzyme called DNA ligase, which is also an important one, okay? All right, so that's the idea of DNA replication. It's a big concept in just a couple of slides. So be aware if you are, I think there is a group that's just going to have just DNA replication. Is Alpha group this time? I don't even know. Somebody has DNA replication just by itself, I'm pretty sure. Be aware. It's a lot of info to cover in what we did like 3 slides on. Right, next we have the central dogma of genetics. This is what we kind of talked about with our viruses, yeah? We had the DNA to RNA to protein. That's the central dogma. This part here, DNA to RNA, is called transcription. And this part's called translation. Now, to transcribe something, if you think of a scribe that's writing something, right? You're probably looking at this thing and just writing down what you need off of it. Like we said, the big book from the Smithsonian with everything in it, going in there and copying down just what we needed. We are being a scribe, right? Easy enough, okay? We're still going from nucleic acid, DNA, nucleic acid, to RNA, nucleic acid. So the original print would be the D, and then now us copying it, we are putting it in the R. This is your handwriting, basically. Next is translation. We're going to take that RNA that you made the copy with, that transcription, that transcript we call it, and you're going to translate it. Because guess what? RNA into protein, that's a completely different other language. We're not nucleic acid anymore. Now we're talking about nucleic acid into protein using amino acids, right? So that's what that's going to be referring to. So that's translation. So we're actually translating it into a different language now. All right. Yeah, it talks about the central dogma is incomplete. I'm not too worried about it. We kind of learned a little bit from our viruses that you can go the other way. Unfortunately, like this is how it works. But this is how we're going to be learning about our genetics. I don't need to get too fancy. All right. So all RNA has to come from DNA, whether it is messenger RNA, transfer RNA, ribosomal RNA, or these other RNAs that I'm not going to get too crazy about, okay? But they do exist, and they usually are there to silence genes. They just like go and like match up with the gene, and they make it stop making products. It's usually that. So anytime it's RNA, it has to come from the DNA originally, right? And if you're messenger RNA, you're going to be read by a ribosome to make your protein. Ribosomes are made up of ribosomal RNA, go figure, and we're going to be using tRNA to bring our new amino acids over. All right, so the gene protein connection, we already kind of know. Proteins ultimately determine your phenotype, whether you have the brown hair, all of that is going to be determined by the proteins that you're expressing. But this can differ from your genotype if you have some sorts of, like I said, cells expressing certain things, or if you have mutations that, suppress certain things. So proteomics is the study of organisms' complete set of expressed proteins. Yes.
Only in viruses,
Yes, yeah, that'll only be in viruses. But yeah, that taught us that like there are at least some cases out there where you can go back from RNA into DNA, which was like defied all logic. We didn't think anything could do that. So that was a new concept, but yeah. Yeah, so your proteins, whatever you're expressing is ultimately what's gonna determine your phenotype. Okay, so let's look at our DNA protein relationship quickly. In DNA, you can separate out your DNA into 3 nucleotides. So let's say this is A, T, C or something, okay? And don't get too picky about this, please, because I'm just putting random things up here. But these will be your triplets. So we can divide it into 3s. We'll call those triplets if they're DNA. Once we've made our copy of our codon and wrote down the information that we need for our gene to take back with us, our mRNA take back with us, these also we can divide them by 3. These are called codons, and this is more important. A codon on mRNA is more important really than the triplets on the DNA. Obviously, the triplets on the DNA dictate what the codons are going to be on the RNA, of course. And you can see it even in the picture, like pretty much, look how this strand of DNA is the exact same structure as this of the RNA. Obviously, we're going to replace the T's in the DNA with U's, uracil, in RNA when we get to that. And yeah, they've got a different sugar, ribose, but still, look, the general code is going to be the exact same. We have a template strand and a coding strand in DNA, and that template strand is what we're making our copy out of. So this would be the template, right? We can just match up, I don't know why it does that, the RNA with the template strand and make our little nucleotides. And it happens to look like this bottom one, which we then would call it the coding. If you're trying to interpret what the coding was going to be for the mRNA, then you can just look at that sequence on the DNA. Okay. And that might be, it's not like one strand is the template strand and one strand is the coding strand. It depends on what gene you're looking at and how it's being expressed, that you could be looking at one side or the other side ultimately of that chromosome at any given point. But for this gene, template encoding, the template is the template to make your messenger RNA. We've made our copy of our messenger RNA, and each codon here, these 3 nucleotides that I keep talking about, each of these leads to one amino acid. And then amino acid links to amino acid, to amino acid, to amino acid, and so on and so forth. Remember, that's going to be our primary structure. Secondary is going to be our alpha helices and our beta sheets, and tertiary is going to be the overall folding of that polypeptide. And come together to make a working one in the quaternary. All right, so transcription and translation. Messenger RNA is important for this. That's going to be a copy of our gene that we're going to be using to read to make the protein. Transfer RNA is important to this. This is a transfer RNA depicted here in this image. Transfer RNA, it looks ridiculous. It looks like a hairpin with three hairpins off of it. And at the top, see where it says amino acid attachment site. At the top, you'll have stuck onto here like whatever, alanine or I don't know, guanine, not guanine, that's the other thing. Alanine or whatever, phenylalanine or whatever else you want, glutamate, a whole bunch, 20 of them. And it'll be carrying one of those depending on what your codon tells it to carry. So you have a whole bunch of tRNAs carrying alanine, a whole bunch carrying phenylalanine and whatever. And that codon calls over the right one that matches to it. If it was ATC, then it would be TAG, for example, or yes, that would match up complementary to it. And it would bring over that amino acid. And that's how it reads it. It's pretty crazy to think about. But they're always going to match up, right? So they have to match up just like the double strand DNA does. So on your mRNA, you'll have your codons, codon 1, codon 2, and codon 3, for example. And this one says AUG on our mRNA. That's always our start codon in almost every organism. So A binds with U, remember that's the equivalent of T. U binds with A, and G binds with C. You see how they're complementary to one another there? That's the anti-codon on the tRNA. So that's how the tRNA is going to know what amino acid to bring over when we get to that part. All right, we also need ribosomes for all this. They're basically the machines that are going to help position everything and hold everything together so we can actually get the work done. They're ribosomal RNA. And then we have other regulatory versions of that, but whatever. Yes.
You said almost. They were start with AG.
Well, it's different organisms usually do that. So yeah. So for example, if we are bacteria versus animals, I'm pretty sure bacteria always start with a version called formyl methionine. So it's like, it's always AG still, but it's like a different version of methionine that they use versus like us. And so there's other organisms out there that like might code a different amino acid altogether at their start, but usually there's a defined start for every, like you're an organism, you're always going to use this, like that. All right, so RNA, remember typically we're going to see it in double, can't speak today. Typically we're going to see it in single-stranded molecule. We can see it forming a helix, even though it's a single strand, just because of the nature of it wanting to do that, because of the backbone, still sugar and phosphate repeating. It does have uracil instead of thymine, so U instead of T. This is just how these chemicals are going to be interacting with one another and due to the temporary aspect of the RNA. And then again, sugar is ribose, not deoxyribose. So we have our messenger RNA, that's our copy of our gene. We have the codons, that's going to be our triplet bases on the messenger RNA. Remember, it's a triplet. If it is DNA, it is codon, if it is RNA. We've already talked about our transfer RNA. That'll have those complementary 3 nucleotides to our codon, and we call that the anticodon. At the bottom of it, I guess they call it a clover leaf. That's nice, isn't it? Yeah, at the bottom of that, so it can match up with the messenger RNA and determines which amino acid's coming along. Ribosomes are made up of ribosomal RNA. I don't know what else to say about this. There is some protein involved in the shape too. That's really all we need to know. There's 2 subunits. They basically come together, a big one and a small one, and they clamp on to the messenger RNA and then just start reading along with it. I say reading, like they're not really doing the reading so much as they're positioning to allow the tRNAs to come in. And then when there's a reaction, they can shift down and a reaction and shift down. I highly recommend watching the video on that because it's way better than I can explain. All right, so first let's talk about transcription and how it starts. We know DNA to RNA, we have to make our messenger RNA first in order to make our proteins. DNA to RNA is called transcription. And guess what? It's really not that difficult of a sequence, thankfully. We have our first step is called initiation. We have RNA polymerase. I bet you can guess what that does. Makes, adds RNA nucleotides. And then we have a promoter sequence. This is the sequence where that RNA polymerase recognizes it can bind and start adding on nucleotides, right? We'll have separation of the strand and start adding on complementary nucleotides basically to the template strand, right? But this is the initiation step. All right, next we have the elongation step, where we start adding our nucleotides, our RNA nucleotides, as we're reading along, and we add them again. Look at that, wouldn't you know it? What is that? 5' to 3'. It's always, okay? Always 5' to 3'. So anyways, we're adding our little messenger RNA nucleotides from the 5' to 3' direction. And eventually we're going to get to a site. We'll say it's the termination site. It's like a stop signal where that RNA polymerase will just like kind of disassociate and we'll be done. So that's how it signals it. That's it for transcription. Okay, let's do questions really quick. I know this is quite a doozy chapter for questions. I am aware. If you have your notes available, I'll allow you guys to look at your notes. It's been a little while since they've done this. Okay. All right, first question. Lauren, what makes DNA replication semi-conservative? What does that mean? Best guess. So remember, conservative is going to be conserving. So it'd be like your template and then conserving that template original. And then having, I don't want to give away too much. So this is referring to, I'll say this, replication. Guys, if you take anything out of this to help you study for this chapter, don't confuse replication and transcription and translation. Those are all completely different things. They are not like, the same thing. So DNA replication. This is, you remember what that is at least?
To make you.
Yeah, right, sure.
The way to keep the same like DNA strands and conserve it.
So yeah, so that's the right thinking. I mean, I'm gonna give you the point for it because you got through it for sure. But so semi-conservative because we're gonna take the original strand, split them apart. They'll each have one old strand and one new strand on the new daughter ones. Okay, so that's what that means by semi-conservative because we'll have one old strand still. We're conserving that strand. Okay, so it's kind of like I always try to tell people, it's hard to explain it, and that's what I don't like about a lot of this genetic stuff. But like if this is your original strand, and you're going to make copies of it, then you'll be splitting this apart, right? Into this. This is like the two strands you just saw up there, right? Now, when you make a copy of them, and yeah, there might be replication forks and all this, but we're not getting into that right here. Okay? They'll be like this: new. New. That makes sense? Yeah. Want to draw it that way? So that's why we say old, new, semi-conservative. Okay. All right. I know this chapter is a lot. Okay. We have Ingrid. During replication, which strand makes the Okazaki fragment? You know the term for it.
I don't know if this would be right.
leading strand?
Okay, so we're in the right area, at least with the concept. So this will be the lagging strand. That's why it's lagging behind, because it has to make the different fragments. Remember, the leading strand is just going to crank through. It doesn't need to make parts, and it can go really fast and just keep going. The lagging strand, that guy will go in parts. Okay, our next question is for Sarah H. Generally speaking, what is the overall purpose of transcription? What does transcription do?
But transcribing the DNA that it's making, so like
Yes,
putting it through.
and into what?
Oh.
It's okay. What do you think it's being made into? Being transcribed into?
DNA.
RNA. So,
RNA,
so
yeah, okay.
so
I was like,
replication-
amino acid, RNA,
so don't-
protein.
yes. So I know it's a lot of terms. Replication, we're literally replicating the DNA. All right, that's just that one. Transcription, now we're turning the DNA into the same message, but it's going to be in a different nucleotide. So right, it's going to be the RNA. But yes, the thinking was good. We're almost there. Yes. All right, what is the function of messenger RNA? We have Tammy. Oh, she's not here, I don't think. Oh, you're really excited. All right. Is Hannah here? She's not here. Or some pulling on people who are just like happen to not be here today. Marissa. Okay. Yeah, so that's basically it, right? So it's making the copy of the gene, the working copy, if we can look at it that way. Okay.
The mRNA, what the, it is pretty much the product of transcription?
Yes.
Okay, I thought so.
All right. I think that's it. Yes, that's it. All right. Guys, people sit down and
I'm a little confused about the tRNA.
the RNAs.
The tRNA, I'm a little confused.
Well, we're gonna get into it here in a minute. So.
Okay, Matt.
All right, so with translation, so now we have our messenger RNA nucleotides are read in codons or groups of three. You want to write really quick up here, so we're going to we will have DNA. We're making copies of this. That's replication. We're going DNA to RNA. This is transcription. So we don't get too lost in this. I know I'm going to be saying these words over and over again, and at least something is here for you guys to know what's happening here. All right. Translation. This is going to be proteins. Remember this, the subunits that we're using for both of these will be nucleotides. These will be ribonucleotides and these are deoxyribonucleotides, right? These guys, what are proteins made of? Amino acids, right? So we're about to add on amino acids to make proteins. So that's why I want to be sure we're on the same page here. Like I said, it's hard to describe stuff that's like action based. So, and this is really like super action based. When I tell you that pretty much everything going on in your body is dictated by proteins, I mean, you're constantly doing this. I mean, it's just always going on. It's not like something that happens once in a while. Okay, so mRNA nucleotides, we read them in groups of three. Those groups of three are called codons. Okay, mRNA has codons. Typically, this is a universal code. And these are all the possible codons that you could have in mRNA based on our A, T, C, G. And remember, T will be replaced by U for our RNA, so A, U, C, G. These are all possible codons. Some of them, many of them actually, code for more than one. Code for, how do I word this? Serine, for example, this is an amino acid, one amino acid. It's coded for by any of these three. Now notice that they're all UC something. UC blah, doesn't matter what, always codes for serine. Now there's a reason for this redundancy. This redundancy is what we call the wobble effect. It means that the tRNA are less picky about the last site on some of these guys. And that's because it's to protect from mutation. If we have mutation, making sure that you're still coding for the right protein. Because if it changed, if you had a mutation, your original genetic information had UCC, and you had a genetic mutation that went to UCG in the offspring, that's still going to code for serine as our amino acid. That's going to be building our protein. And the protein is what does the work and actually determines your phenotype and everything, right? So we don't care about that mutation. We're actually going to talk about that in a minute too. But you can see here there's a lot of overlap. There's only 2 for phenylalanine, 2 for leucine, and there is 1 for start. The stops are kind of trickled throughout. Stop codons are the 3 that do not code for anything. They will basically, they don't bring along any amino acid or anything like that. They're empty and they don't have any signal. And that's what causes the ribosome to dissociate when we get to that, because it says, oh, this isn't a real thing, and then dissociate. So that's why that's like that. Yes. Okay. So here's a better picture than what I'm drawing. Here are your DNA triplets. Remember, we have a coding strand and a template strand, and your messenger RNA will be the same code essentially as your coding strand, okay? And we use the template strand as the template to make it. So here's our messenger RNA. It reads AUG, CUG, ACU, ACG, always in 3s. These are our codons. Next are our tRNA anticodons. So if you are AUG, your anticodon must read UAC, which is what this one does, right? And so on and so forth. So what happens is you get a tRNA that has the opposite of your codon, called the anticodon. It has the anticodon that matches your mRNA codon. And when it does, that signals to add on its attached amino acid. Okay? Because each of these, when we say, oh, these things code for serine, when I say UCC codes for serine, I'm saying your mRNA says UCC in it, your anticodon says AGG on it, and then signals to attach serine. That's what's really going on here. Okay. So your genetic code dictating which amino acids. So now down here we have methionine. On this one we had leucine, and we had 2 threonines. Look, these are both threonines, even though they were coded for by different sequences. It just turns out, hey, that's one of those overlapping sequences. Just worked out that way. That person had that in their genes. So same amino acid, different codon, different anticodon as well. All right, cool. All right, so we're going to bring all the elements we need in order for translation to happen. Remember how when we went through transcription, all right, to make our messenger RNA, our working copy, that we had initiation, elongation, and termination. Luckily, that's all that's happening here as well. Same steps, all right? So initiation, we're going to be, okay, yeah, let's go over the terms, I guess. The start codon is where we're going to start everything off. It's always AUG. There are other organisms that don't necessarily have the same, but again, like I said, we need to worry about this. Stop codons are what we call nonsense codons because they don't have a corresponding tRNA, which causes it to terminate. Okay, so that's these guys down here. And then translocation is the term that we use for the ribosome shifting down to the next codon. And it'll shift on down to the next codon, and then shift on down to the next codon, and keep on reading that. You can have in bacteria are the coolest, by the way. They can, you know how we have to make our messenger RNA from our DNA, right? And that working copy, we have to take it out of our nucleus and into the cytoplasm, or rough ER, and then make our protein there. Bacteria don't have a nucleus. So they can literally start making their messenger RNA, and we can bind on our ribosomes before it's even done being made. And so that's crazy to think about. And you can have multiple ribosomes just reading down right next to each other. The next one comes on, the next one comes on, and you can use one messenger RNA to make multiple copies of protein. So you can have all of that going on before you're even done making your messenger RNA if you're in bacteria. Okay, so this is where I absolutely have to advocate you guys watching the video. How am I supposed to explain this one? So we have our tRNAs. They look like little T's upside down, and they're attached to their respective amino acids. So we always start with AUG. Our AUG on our messenger RNA causes the ribosome to clamp down and bring in our first tRNA that has methionine or F-methionine, whichever organism you are. Now, that's great. In the next little spot, we can get our next codon matching up with the next tRNA, right? So that's great. So we have amino acids and they're hanging out on the tRNAs. How does that help you at all, right? So we're going to create peptide bonds. That's what the that's what the ribosome is there kind of helping foster. The peptide bonds between those two amino acids you just brought in, bound to one another. And then we can kick out the first guy, move the second guy over into the first spot, bring the next guy into the next tRNA position, anticodon to codon, with its amino acid, link it on, kick out the first guy, push that guy over, and so on and so forth. And that's basically how this is going to work. Like I said, if watching the video, I don't have it in here, wouldn't let me put the video in here. I don't know why, but I might have it somewhere. Let's see how much time we have. Not really that much time. Okay, but yes. So this is how it can look as well. Even though we can't necessarily, we don't have the ability to do this. The bacteria do. So you see, we're making our mRNA off of our DNA actively there, and have all these ribosomes lined up, just cranking out protein, shifting down, moving over for the next tRNA, linked to the next amino acid, and it's going to get linked to each other, and then we shift down again, and all of that over and over again. Okay. All right. So COVID mRNA vaccines, they contain viral mRNA. It codes for surface protein of the virus. Once we inject it into the arms, our cells will use their own machinery to make that protein, the COVID protein, and express it. And then our immune system will see that as foreign and attack it. So, you know, basically hijacked the ability of your body to create protein, and then also, depending on your immune system, to react to it as foreign. So it's a pretty cool concept. So that's the first time that we've ever had actual vaccines put into use based on that. We've had plenty of trials with them and everything like that, but yeah, this first time we had that in humans, and it was effective. Changing the face of medicine. All right, so prokaryotic and eukaryotic, I don't really need to get, I don't care really that much, other than you can do both at the same time for bacteria. The thing that sucks about eukaryotes is we have exons and introns. Okay? Introns are intervening sequencing, introns, sequences of bases that don't code for anything. So you come over, you make a copy of your gene, and you're writing down your copy, and in the middle, you're just writing bit, bap, boop, bap, bap, in the middle of whatever you're doing, right? And now you've got to go home, and you're like, well, great, you just wrote bit, bap, boop, bap, bap, and you have to like basically cut it out. So that's what this is. We're cutting out extraneous information. We'll cut out the introns and splice together the exons. Your body does this on the mRNA level. We'll subject it to the spliceosomes and merge together those exons. So you're either intron or your exon in eukaryotes. And so we'll cut out the introns, merge together the exons. I don't have a good way of remembering this. If you guys think of one, please share with each other, because this can get confusing. You forget which one is which. All right, the spliceosome, I'm not really going to test you over it, but that's just the area where we're going to be putting those exons together and cutting out the introns. All right, this is one of the worst, hardest topics for everybody, okay? Operons. Bacteria and archaea can do this thing where they can regulate a whole bunch of genes all at once in like one long strand as a single unit. We don't do that. We have to have separate for each of our genes. But if they're all involved in, you know, breaking down lactose or making arginine or something, then they can turn on that one thing and it gets all of the genes that you need that's related to it. So that's called an operon. They can be inducible or repressible. Inducible operons, basically, we're going to have genes that we need to turn on. They're not on usually. It doesn't benefit the organism for it to be on unless it needs it. So, like we're saying, you need to break down lactose. You don't usually see lactose as bacteria. This inducible operon, lactose comes along and it basically induces these genes to turn on. And this is how it's going to work. Okay, this is an operon. Oh, okay. Why do you have me introduce both of them and then go over both? No, okay. The lactose, I don't like that at all. Lactose operon is our inducible one. It's called lactose operon, the lac operon. We have the promoter where your RNA, we already know the promoter. We've already met the promoter. That's where your RNA polymerase is going to bind onto and make your friggin' mRNA. Then we have an operator. The operator is where operons will bind their repressor protein and block your promoter, your RNA polymerase. So it can't do its thing, okay? So we have a repressor protein, it says allosteric. Basically, it binds two binding sites, one for the operator sequence and one for lactose. So what happens is lactose comes in, it's your first time eating the lactose in like years, I don't know. And then it comes in and binds with your repressor protein. The repressor protein, anytime proteins bind with another protein, it typically causes them to change shape. Okay, so it's like a chemical interaction. Now we have maybe more hydrophobic interacting with hydrophobic than we like, or something like that. So we'll shift our shape. So we see lactose, we bind with it, we shift our shape, and that now allows us to bind onto that operator region and stuff like that, or come off of the operator region in this case. So now we're going to shift our shape and come off of that operator region and let the RNA polymerase go through. I hope there's a picture of this because I don't like explaining it. Okay, good. Okay, so it's locked in place. RNA polymerase normally comes down and it can't move because that brown horseshoe looking thing is the repressor. Can't go typically, we're turned off. So this is off. This is how it normally is. We don't need always to make these genes. Now we have lactose. It's our inducer because it's inducible. We're going to induce it to turn on these genes. Lactose comes in, it binds with our horseshoe guy, it changes its shape. You see how it goes like a clothespin almost in their image here? And it moves away. So now RNA polymerase can come and make products and we can get the proteins made. When we run out of lactose, that loose association with the repressor protein, it can go bind on back again and block it all over again. Don't have it. This is an inducible. If you have to, remember this, it's inducible because you can induce it. Like if you can't induce labor, there's labor, you have to have somebody be pregnant first, right? You can't induce labor without that. So it's inducible. And we know what induce means. Okay, that's why I'm saying this. And we know what repress means. It means this is an operon that can be repressed. I say this because everybody gets this one confused. All right, so repressible operons are on. They're going to turn off when we don't need the thing anymore. And the excess of whatever you're making is usually our repressor. So we have our operator. Both of them have operators. Cool. That's here. So normally RNA polymerase comes along and we're making our stuff. Cool. We're making our protein. Cool. Great. What are we making as our after our proteins? Our proteins are helping us make arginine, which is an amino acid. We're making a whole bunch of arginine. Isn't that nice? That's what we're always going to be doing, except until we have too much of it. When we have too much, if the body isn't using up the product enough, then the product will build up. And then it'll go interact with the repressor. So if the body isn't using arginine in metabolism, then it'll build up and bind with the repressor. So let's say that's what's happening here. These are the ones we weren't using. I know not as active anymore. We don't need to make as many copies of the cells or something. It'll bind with the repressor. And then just like before, that will bind on the operator. So the RNA polymerase can't keep making those genes. So it's literally like the opposite situation. You guys see how that works? Always the operator is the spot where the repressor is going to work. They both have repressors. It's just a matter of whether you're going to induce production, which means removing the repressor, or you're going to repress this, and that means putting on a repressor. Okay, so that's what this is talking about. Understanding the mechanisms by which that is controlled helps this be clearer if you can work it out in your own mind. I can only explain it so many times. So I would encourage, if you can, watch a video over that. Okay. DNA recombination is dealing with any organism containing and expressing genes from another organism at all, ever. Horizontal gene transfer is whenever the bacteria transfer the genes back and forth. We mentioned this with the sex pilus, right? That was something that we talked about that bacteria can do. with the sex pilots, they're going to transfer their DNA between one another. That's called conjugation. They can also transfer their stuff between one another by, spreading their viruses. So it's called transduction. Remember we learned about lysogenic conversion. That's how that would happen. And then we also have something called transformation, which means picking up DNA from outside of you. It could be other dead cells that died recently, picking up their DNA and incorporating it into yours. We also have plasmids. These are extra little circular bits of DNA that can contain helpful genes. They're not required, but they are useful. So here's the 3 that I just went over. It's a good table to review that if you're interested. Okay, we already talked about these. I don't need to go over too much. Transduction, the viruses. Okay, let's go through the mutations. This is any change to a nucleotide sequence. Any change, whether you are still making the same amino acid or not, it's still considered a mutation if it changed. Okay, so even a single point. It can be spontaneous, could be random, just because of mistakes made in making copies, or it can be induced due to chemical or physical agents, which we call mutagens, like UV light, for example. We have missense, nonsense, silent mutations. Then we have back mutations, which aren't as important. But missense, this is any code that leads to a different amino acid. Okay. Originally you coded for serine, now you're coding for phenylalanine because of your change. That's a missense mutation. This is all going to be dependent on what your product is. Nonsense mutations, anytime we change our codon into a stop codon. We're nonsense because we're not making any more amino acid. Okay. Silent mutations were the ones that we talked about earlier. You can have a change in your nucleotide sequence, but still have the same exact amino acid. That's a silent mutation. The serine, yeah, how it can have like the 4 different.
Was like
You're okay.
2 of the same one at the end.
Yes. So these, I don't want to go back. There's we have a million clicks back. I don't want to stay for too much longer. But yes. So your codons, right? You can have serine be coded for by 4 different codons, for example. If you had a change that changed it, and it still codes for serine, that's silent mutation, but it is a mutation. Then we have back mutations, where you had some sort of mutation that reverts it back to the original form, what we call the wild type. So this is going through a few of the different kinds of mutations you can have, where if you have this sequence in your DNA, which would lead to this copy in the RNA, leading to a code that is 1, 2, 3, this glutamate, but then you have a change in it, CTA, which it was GAU instead of A, and now it's this instead. So you might not think, you might be looking at these and like, who cares? Glue to ASP. Some of these guys are very polar, some of these guys are very hydrophobic, and that dictates how your whole protein could be behaving. So yeah. All right. Frame shift mutations, if you have 1 or 2 bases deleted or added, it shifts the whole frame. It's like taking a book and all of the letters where they are, keep them the same size of words, but take out 1 letter and then shift everything based on that 1 letter how would you read that? It would be a hot mess. You could probably figure it out, but you know what I mean? If you're like literally as a chemical reader trying to read that would be a big problem. That's frame shift mutations. It can be absolutely devastating to the organism. But yeah. Anyways, all right, so I think it's the last one, right? The Ames test, this is basically a test to determine carcinogenic potential of things. We usually use like some kind of bacteria like salmonella, and then take away its ability to make histidine, and it has to grow on this plate that we add histidine into. And if it mutates, then it can now grow without us adding it. That's a mutagen. So that's what it thinks about. So that's the Ames test. We're no questions this time. We're just gonna go to the lab. Guess we got some in. I think we'll get more in on chapter 8. It's kind of smaller, shorter.